What if the universe's very first living organism wasn't something that emerged from Earth's primordial oceans, but instead began somewhere out among the stars? Picture this, a single, microscopic entity awakening in the vast cosmic darkness, the pioneer of all biological existence. This isn't just speculation, it's one of the most profound mysteries science has ever attempted to solve, and it leads us down a path filled with extraordinary possibilities that challenge everything we thought we knew about life itself. Today, we're embarking on a journey to explore 30 incredible theories about how life might have begun, not just on our planet, but potentially anywhere across the cosmos. From lightning sparking the first amino acids in ancient Earth's atmosphere, to microorganisms hitchhiking on asteroids, to the mind-bending idea that life could have emerged in the hearts of distant nebulas, each theory opens a door to a new way of understanding our place in the universe. This isn't just about biology or chemistry, it's about the grand story of existence and how something as fragile as life could arise in a universe that seems, at first glance, overwhelmingly hostile to it. So settle in, because we're about to dive deep into the science, the speculation, and the sheer wonder of it all. Whether you're a curious casual viewer or someone who's spent nights staring up at the stars and wondering how it all began, this exploration is for you. By the time we're done, you'll have a new appreciation for the resilience of life, the creativity of scientific inquiry, and the breathtaking possibilities that await us as we continue to unravel the mysteries of the cosmos. Let's begin. Our first stop on this journey takes us far beyond our own world to the vast and silent expanse between the stars. It's a theory that suggests life on Earth might not be a native product but an immigrant, a cosmic seed that traveled across the galaxy to find a fertile new home. This is the concept of Panspermia, and it fundamentally reshapes our understanding of life's origins. Instead of a singular genesis event in a primordial pond, life could be a constant, widespread phenomenon in the universe, simply hitching rides on celestial taxis like comets and asteroids to spread from one star system to another. The idea isn't as new as you might think. While modern versions are grounded in astrophysics, the core concept dates back to the ancient Greek philosopher Anaxagoras, who used the actual word panspermia to propose that life's seeds were present throughout the universe. But it was revitalized in the 20th century by some formidable scientific minds. The renowned astronomers Sir Fred Hoyle and Professor Chandra Wickramasinghe became its most famous modern proponents. They argued that the raw ingredients of life, and perhaps even dormant microorganisms, are abundant in interstellar dust clouds and are constantly raining down on planets. They even controversially suggested that some pandemic viruses could have extraterrestrial origins, arriving on cometary debris. But is there any tangible evidence to support this star-spanning notion? Surprisingly, yes. We've discovered that some meteorites that have crash-landed on Earth are rich in organic compounds. The Murchison meteorite, which fell in Australia in 1969, is a famous example. Analysis has shown it contains a plethora of amino acids, the very building blocks of proteins and life as we know it. This proves that the fundamental chemistry for life can and does form in the extreme environment of space. Furthermore, experiments have shown that certain hardy bacteria, like Deinococcus radiodurans, can survive the incredible shock, radiation, and cold of space travel, especially if sheltered within the interior of a rocky asteroid. This lends credibility to the idea that life could endure an interstellar journey, dormant but still viable, until it crash lands on a welcoming young planet like Earth billions of years ago. Yet while Panspermia offers a thrilling cosmic delivery service, it still leaves one massive question unanswered. Where and how was the very first package assembled? If life didn't start here, how did it start anywhere? To answer that, we must journey from the cold vacuum of space to the very opposite extreme, the crushing, superheated depths of a young planet's oceans. Here, at the very bottom of the sea, lies a theory that doesn't require a celestial delivery at all. It suggests that life is a natural, almost inevitable consequence of planetary geology, emerging from the fiery, chemical-rich hearts of deep-sea hydrothermal vents. Imagine the early Earth, a violent and turbulent world still cooling from its formation. The ocean floor is a dynamic, fractured landscape, and from these cracks in the crust, incredibly hot, mineral-rich water spews forth into the frigid sea. These are hydrothermal vents, and they come in two main types that scientists believe could have been life's original kitchens. The first are the dramatic black smokers, towering chimneys billowing out dark clouds of superheated water saturated with iron sulfide and other metals. They look like something from another world, and in a way they are, a glimpse into our planet's most ancient and powerful processes. But perhaps even more promising are the less dramatic but chemically fascinating alkaline vents, also known as white smokers. Unlike their black, acidic cousins, these vents release warm, alkaline fluid rich in hydrogen. The key to their potential lies in their very structure. They form porous, rocky formations full of tiny, interconnected compartments and channels, almost like a natural labyrinth of microscopic rooms. These nooks and crannies would have served as the perfect incubators, concentrating the simple organic molecules that were either delivered by meteorites or formed in the early atmosphere and washed into the sea. Within these porous walls, an incredible chemical dance could have begun. The vent fluid, rich in hydrogen and other electron donors, would interact with the more acidic ocean water, creating a natural proton gradient, a fundamental form of energy not unlike the one that powers every single living cell today. This energy could have driven the formation of more complex molecules. The mineral surfaces, particularly certain types of iron and sulfur compounds, could have acted as early catalysts, accelerating reactions that eventually led to self-replicating systems and the first primitive metabolisms. It's a compelling picture, not a lightning strike in a stagnant pond, but a slow, steady, and protected genesis, fueled by the planet's own inner heat and chemistry, brewing the first life in its deep-sea laboratories. But if the deep-sea vent theory suggests a protected, geothermal genesis, another famous idea places life's cradle in the open air under stormy skies. We now travel from the ocean's abyssal plains to its sunlit surface and the atmosphere above, to revisit one of the most iconic origin stories ever proposed, the primordial soup theory. This concept paints a picture of Earth's early oceans not as a barren wasteland, but as a warm, nutrient-rich broth, a literal soup simmering with the organic compounds that would one day become the ingredients of life itself. The foundation for this idea was laid independently by two brilliant scientists in the 1920s, the Russian biochemist Alexander Oparin and the British geneticist J.B.S. Haldane. They reasoned that Earth's primitive atmosphere, which they believed was very different from our oxygen-rich air of today, was the key. They proposed it was a reducing atmosphere, rich in gases like methane, ammonia, hydrogen, and water vapor. With no free oxygen to break them down, simple organic molecules could form freely. Haldane famously suggested that the ancient oceans reached a consistency akin to a hot dilute soup, where ultraviolet light from the sun, atmospheric lightning, and volcanic heat provided the energy to cook these gases into ever more complex compounds. Over immense stretches of time, these compounds concentrated, eventually organizing into the first self-replicating structures. This compelling hypothesis remained just a theory until 3 decades later, when a young graduate student named Stanley Miller, working under his advisor Harold Urey at the University of Chicago, decided to put it to the test. Their now legendary experiment was stunningly simple in concept yet profound in its implications. They built a closed glass apparatus designed to mimic the conditions of early Earth. One flask held water representing the ancient ocean, which was heated to produce vapor. This vapor circulated into another chamber filled with the proposed primordial gases, methane, ammonia, and hydrogen. In this atmosphere, they simulated lightning storms with continuous electrical sparks from electrodes. The resulting mixture was then cooled, causing the water to condense and trickle back into the first flask, representing rain cycling chemicals back into the ocean. After just 1 week of running this continuous cycle, something extraordinary happened. The water in the flask, which had started clear, turned a murky, reddish-brown. When Miller and Urey analyzed this primordial soup, they found it was teeming with organic compounds, including several different amino acids, the very building blocks of proteins. It was a monumental achievement. For the first time, someone had taken inorganic matter and, using nothing but conditions presumed to exist on the early Earth, created the fundamental ingredients of life. The experiment sent shockwaves through the scientific community and seemed to provide powerful, tangible evidence that life could have emerged from a chemical soup right here on our planet. However, as with any good scientific story, the plot has thickened considerably since that groundbreaking discovery. Modern geology and astronomy have led to serious criticisms and necessary updates to the classic primordial soup model. One of the biggest challenges is the composition of the early atmosphere itself. Scientists now believe it was likely less reducing than the mixture of methane and ammonia used by Miller and Urey. Instead, it was probably dominated by carbon dioxide and nitrogen, gases that are far less reactive and much less efficient at producing the rich array of organic molecules seen in the original experiment. When later researchers replicated the Miller-Urey experiment with this more neutral gas mixture, the yields of amino acids were dramatically lower. This doesn't mean the theory is completely wrong, it has simply evolved. While the atmosphere might not have been the primary kitchen, other energy sources could have powered the soup. Volcanic eruptions, intense ultraviolet radiation streaming through an ozone-less sky, and even the impact shockwaves from constant meteorite bombardment could have provided the necessary spark. Furthermore, the soup itself may not have been a uniform mixture across a vast ocean. Tidal pools, evaporating lagoons, or the surfaces of clay minerals could have served as crucial concentrating mechanisms, gathering the sparse organic molecules from the large volume of the ocean into tiny, rich pools where chemistry could truly take off. So, while the classic image of a lightning bolt jump-starting life in a global ocean might be an oversimplification, the core idea that Earth's surface environment was a crucible for prebiotic chemistry remains a powerful and essential piece of the puzzle. Yet, even as we consider the power of hydrothermal vents and the promise of a primordial soup, we run into a classic chicken and egg problem that has long perplexed origin of life researchers. Which came first, metabolism, the complex network of chemical reactions that harnesses energy, or genetics, the system for storing and replicating information? A protein enzyme is needed to replicate DNA, but the instructions to build that very enzyme are stored within the DNA itself. It's a frustratingly perfect closed loop. How could such an interdependent system have ever bootstrapped itself into existence from simple chemistry? This is where our fourth theory presents an elegant and revolutionary solution, suggesting that the very first life form wasn't a cell at all, but a molecule, one that could perform both jobs at once. Welcome to the RNA world hypothesis, a concept that proposes a stage in Earth's early history where RNA reigned supreme, acting as both the genetic blueprint and the molecular machine, long before DNA or proteins ever entered the picture. So what makes RNA so special? To understand its unique qualifications, we need to look at its structure. Like its more famous cousin DNA, RNA is a nucleic acid, a long chain made up of building blocks called nucleotides. This allows it to store genetic information in its sequence, much like a string of code. But RNA has a crucial talent that double-stranded DNA lacks. It can fold into intricate, three-dimensional shapes. This folding allows certain RNA molecules to act as catalysts, speeding up specific chemical reactions. These catalytic RNA molecules are called ribozymes, and their discovery was the key piece of evidence that propelled the RNA world hypothesis from a curious idea into a leading theory. The discovery of the first ribozyme in the 1980s by Thomas Setch and Sidney Altman, which earned them a Nobel Prize, was a genuine shock to the scientific community. It proved that a single molecule could indeed be a master of two trades, an information carrier and a chemical catalyst. This dual capability neatly sidesteps the chicken and egg paradox. In an RNA world, the same molecule that carries the genetic instructions for replication can also fold up and perform the act of replication itself. A single ribozyme could, in theory, copy its own sequence, making it both the software and the hardware, a self-replicating entity that could undergo evolution by natural selection. This would represent the very dawn of Darwinian evolution, all housed within a single, versatile type of molecule. Imagine shallow pools on the early Earth, warmed by a young sun, rich with nucleotides formed by atmospheric chemistry or delivered by meteorites. Within this molecular soup, short strands of RNA would randomly form. Most would be inert, but by sheer chance, one might fold into a shape that allows it to weakly link nucleotides together, perhaps copying a small part of another strand. This crude replication wouldn't be perfect. Errors or mutations would creep in. Some of these new variants might be even slightly better at replicating, and they would begin to outcompete their neighbors. This is the spark of evolution, selection, and competition at a molecular level, driving the improvement of these first replicators over millions of years. Eventually, these RNA molecules might have evolved to build protective fatty membranes around themselves, becoming the first protocells, and later learn to code for proteins, which are far more efficient catalysts, leading to the DNA-based world we live in today. While the RNA world is a beautifully elegant theory, it's not without its own formidable challenges. The most significant hurdle is the sheer difficulty of forming long strands of RNA abiotically without the help of modern cellular machinery. RNA's nucleotides are complex molecules themselves that are tricky to form under prebiotic conditions. Furthermore, RNA is notoriously unstable. It degrades quickly in water, and it's hard to imagine how long, functional sequences could have been assembled and maintained in the chaotic environment of the early Earth. Scientists are actively working on these puzzles, exploring how mineral surfaces like clay might have protected and concentrated nucleotides, or how cycles of wetting and drying in tidal pools could have driven the formation of longer chains. Despite the challenges, the RNA world remains one of the most compelling frameworks we have for understanding how the immense gap between non-living chemistry and true genetics could have been bridged. But perhaps the elegant RNA world still relies on a molecule too complex to have been the true starting pistol for life. If prebiotic chemistry struggled to assemble RNA's intricate nucleotides, maybe the very first steps toward life didn't use organic molecules at all. This leads us to a truly radical and ingenious fifth theory, one that suggests life's original architect wasn't a carbon-based compound, but simple, common, unassuming mud. This is the clay catalyst theory, a concept that completely reimagines the cradle of life, proposing that the intricate patterns in minerals provided the first blueprint for replication and evolution. The theory was most famously championed by the Scottish chemist and molecular biologist Alexander Graham Cairns-Smith in the 1980s. His revolutionary idea began with a simple observation. The genetic code in our cells is at its heart a system for storing and copying information. But what if, long before nucleic acids like RNA or DNA existed, there was another, much simpler system that could also store information and be subject to evolution? Cairns-Smith looked at the humble clay minerals forming at the bottoms of rivers, lakes, and shallow seas and saw a perfect candidate. Certain clay minerals, like montmorillonite, have a fascinating crystalline structure. They form in stacked, repeating layers, but these layers aren't perfect. As the clay crystals grow, defects and irregularities, tiny misalignments, substitutions of one atom for another can occur. Crucially, when a crystal breaks and a new one starts to grow on its surface, it often copies these defects. The new crystal inherits the mistakes of the old one. In the most basic sense, this is a form of information transfer, a primitive kind of replication where the structure of one crystal influences the structure of the next. Cairns Smith argued that this process could have been the foundation for a very early, mineral-based form of natural selection. Crystals with certain defect patterns might grow faster or be more stable in a particular environment, outcompeting other crystal types. For millions of years, this non-organic evolution could have slowly refined these crystalline genes. But how do we jump from evolving crystals to the first living cells? This is where the theory becomes particularly compelling. These clay minerals aren't passive observers, they are chemically active. Their surfaces have a natural electrical charge that can attract and concentrate simple organic molecules from the surrounding water, holding them in place and organizing them into orderly patterns. Imagine a clay crystal acting like a magnetic template or a sticky workbench. It could have gathered amino acids and other building blocks, lining them up in specific arrangements that facilitated chemical reactions between them, eventually leading to the formation of more complex chains like proteins or nucleotides. In this scenario, the clay isn't just a stage, it's an active director orchestrating the chemistry that would eventually lead to organic life. Over immense stretches of time, the organic molecules became sophisticated enough to form their own self-replicating systems, perhaps based on RNA. Once these organic systems became robust and efficient, they effectively took over, bootstrapping themselves off the mineral scaffolding that had nurtured their early development. The clay-based life became obsolete, and the carbon-based life we know began its own evolutionary journey, leaving its mineral ancestors behind in the mud. While the idea of life emerging from clay presents a fascinating shift from organic to mineral beginnings, another theory takes us back to the deep sea, but with a radically different chemical twist. This hypothesis suggests that life didn't just find shelter near hydrothermal vents. It was actually born from the very minerals that form those vents, in a process that created a metabolism before genetics. This is the iron-sulfur world theory, a brilliant and compelling concept proposed in the 1980s by the German chemist and patent lawyer Günther Wechtersheuser. Wechtersheuser's background is key here. His experience with patents gave him a unique perspective on complex systems and their origins. He approached the problem of life's beginning not by asking about its first information molecule, but by asking about its first source of energy. His central idea was that life began not as a self-replicating molecule, but as a self-sustaining network of chemical reactions, a primitive metabolism that could harness energy and build complexity from simple ingredients. He called this concept a metabolism-first theory, standing in stark contrast to the genetics-first view of the RNA world. So, where could such a thing get started? Wechtersheuser looked to the same deep-sea hydrothermal vents we discussed earlier, but he focused on a very specific and common mineral found there, pyrite, also known as fool's gold. Pyrite has a remarkable property. It forms from iron and hydrogen sulfide in a reaction that releases energy. Wechtersheuser proposed that the smooth, positively charged surface of a growing pyrite crystal could have served as the perfect stage for the origin of life. This wasn't just a random place for chemistry to happen. The surface itself was the catalyst and the power source. Imagine a tiny, bubbling hot spring on the ocean floor billions of years ago. A slick surface of pyrite is forming, and its positive electrical charge acts like a magnetic trap, attracting negatively charged molecules from the vent fluids and seawater, such as carbon dioxide and other simple compounds. Held firmly against this mineral canvas, these molecules are pushed into close proximity, and the energy released from the pyrite formation itself provides the spark needed to drive reactions between them. This process, known as surface metabolism, could have built increasingly complex carbon-based molecules right there on the solid rock surface. In this view, the first life was essentially a two-dimensional chemical film, a living rust growing on a crystal of fool's gold, feeding directly off the geochemical energy of the planet. The elegance of this theory is that it solves several major problems at once. It provides a continuous and reliable energy source, the formation of pyrite, which is far more dependable than random lightning strikes or ultraviolet light. The mineral surface also offers organization, holding the developing molecules in place and preventing them from simply diffusing away into the vast ocean, effectively concentrating them into a tiny laboratory. Furthermore, this system could theoretically develop a form of natural selection, a patch of this chemical film that happened to catalyze a more efficient set of reactions would grow faster, outcompeting other patches and eventually evolving into a more sophisticated metabolic network. Over eons, this surface-bound chemistry could have eventually produced organic molecules complex enough to form protective membranes and self-replicating molecules, allowing life to peel itself off the rock and venture out into the surrounding environment. While the iron-sulfur world is a powerful and intellectually satisfying model, it is not without its own questions. Scientists have struggled to experimentally demonstrate some of the key chemical steps proposed to occur on the pyrite surface. For instance, the crucial reaction that fixes carbon dioxide into organic molecules using this energy has proven difficult to replicate under realistic conditions. Nonetheless, Wechterscheus's core idea that life's incessant drive for energy found its first expression in the simple chemistry of rocks and minerals has had a profound impact on the field, reminding us that the line between the living and the non-living might be far more blurred than we ever imagined. But if we're to truly extend the idea of panspermia to its absolute limit, we arrive at a concept so vast it challenges the very notion of a beginning. This is the theory of cosmic ancestry, which posits that life did not just hitch a ride on a few asteroids. It is a fundamental and eternal property of the universe itself. In this view, life has no single point of origin because it has always existed, woven into the fabric of the cosmos, with its distribution and evolution being an ongoing process that spans galaxies and eons. It suggests that the seeds of life, or perhaps even fully formed microorganisms, are as much a part of the universe as hydrogen atoms or cosmic dust, perpetually spread by the gravitational mechanics of star systems and the wanderings of comets. The implications of this idea are staggering, fundamentally reshaping our understanding of life's timeline and its potential prevalence. If life is eternal, then its age is effectively the age of the universe itself, some 13.8 billion years. This vast head start means that life elsewhere could be billions of years more advanced than anything on Earth, having had immeasurably more time to evolve, diversify, and perhaps even learn to engineer its own spread across the cosmos. It transforms the universe from a mostly barren collection of matter into a teeming, living entity, where habitable planets are not just rare oases but fertile soil waiting for seeds that are constantly raining down upon them. The galaxy becomes less of a cold, empty space and more of an interconnected ecosystem, with comets and asteroids acting as pollinators carrying the genetic material of life from one stellar flower to another. This perspective also offers a potential solution to one of the most persistent puzzles in the origin of life field: the seemingly impossibly short window between Earth cooling enough to become habitable and the first evidence of life appearing in the fossil record. If life is not something that must painfully bootstrap itself from non-living chemistry on every single planet, but is instead a pre-existing condition that arrives ready-made, then it explains how life on Earth could have taken hold so astonishingly quickly. The planet simply became ready for tenants, and those tenants who had been waiting in the cosmic real estate agency for billions of years immediately moved in. This doesn't just make life on other worlds likely, it makes it almost inevitable, suggesting that anywhere the conditions are even marginally suitable, the ancient, eternal spores of life will eventually find a way to take root and thrive. Yet for all its cosmic grandeur, the theory of cosmic ancestry still treats the spread of life as a blind, natural process, no more deliberate than the wind carrying a dandelion's seeds. But what if it wasn't blind at all? This brings us to one of the most provocative and debated ideas on our list, the theory of directed panspermia, which proposes that life on Earth was no accident of celestial mechanics, but a deliberate act of biological seeding by an advanced extraterrestrial civilization. Imagine not a random meteorite carrying dormant microbes, but a sleek, targeted probe designed to breach a planet's atmosphere and deliberately release a genetic starter kit into a promising new world. In this scenario, we are not just distant relatives to potential life elsewhere in the galaxy. We might be the direct product of an intentional experiment, a purposeful act of creation from beyond our star. This astonishing concept was brought into the scientific mainstream by none other than Francis Crick, the Nobel Prize-winning co-discoverer of the structure of DNA. Alongside British chemist Leslie Orgel, Crick published a paper on the idea in 1973. It's crucial to understand their reasoning. They weren't invoking ancient astronauts or UFO mythology. Instead, they were grappling with a genuine scientific puzzle they called the double origin problem. They argued that the universal genetic code, the specific, complex way DNA instructions are translated into proteins, appears to be a frozen accident. It works, but it seems arbitrary. There's no obvious chemical reason why it had to be that specific code and not a slightly different one. If life had originated independently on Earth, they reasoned, we might expect to find evidence of different, competing genetic codes in the most ancient organisms. But we don't. All life on Earth, from bacteria to blue whales, uses the same fundamental code. To Crick and Orgel, this suggested a common origin that was simply too efficient, too uniform to have emerged randomly from the primordial chaos. A directed seeding from a single source, they argued, would neatly explain this puzzling uniformity. The ethical and philosophical questions this theory unleashes are profound and unsettling. If true, it would completely redefine our place in the universe and our understanding of our own origins. Are we the children of a benevolent, God-like civilization that sought to spread the gift of life throughout a barren cosmos? Or are we merely the subjects of a grand, impersonal scientific experiment, thriving in a planetary petri dish while our creators observe from a distance, utterly indifferent to our fate? This idea touches on the deepest human questions about purpose, creation, and divinity, blurring the lines between science and religion in a way few other theories dare to. It suggests that the search for extraterrestrial intelligence is not just a search for our peers, but potentially a search for our parents. Of course, directed panspermia faces immense scientific hurdles, the most significant being the total lack of any direct evidence. Critics rightly point out that it doesn't actually solve the problem of life's ultimate origins. It merely pushes it back in time and out into space, forcing us to ask how that alien civilization came to be. Furthermore, the challenges of interstellar travel, even for an advanced species, are staggering. Protecting fragile biological material against cosmic radiation for million-year journeys between stars is a problem of almost unimaginable difficulty. Yet for all its speculative nature, the theory serves a valuable purpose. It forces us to think on a grand galactic scale and challenges our Earth-centric assumptions. It reminds us that the universe is old enough and vast enough for possibilities that seem like science fiction to us, and that the ultimate answer to the mystery of life's beginning might be even more extraordinary than we have yet dared to dream. While we've explored how metabolism or genetics might have emerged, another fundamental question remains. How do these processes become contained and protected from the chaotic outside world? This brings us to a theory that focuses on life's very first walls, suggesting that the containers themselves may have been the cornerstone of life's origin. Welcome to the lipid world hypothesis, a fascinating idea that places fatty molecules, not genes or proteins, at the very center of the story, proposing that the first step toward life was the spontaneous formation of tiny, self-organizing bubbles. Think of a cell as a bustling city. It needs government and industry, the genetic code and metabolic machinery, but it also desperately needs a city wall. Without a barrier to separate the organized interior from the disordered exterior, all the delicate chemistry of life would simply diffuse away into the vast ocean, becoming meaningless noise. The lipid world hypothesis argues that these walls, made of fatty molecules called lipids, weren't a late addition to already functioning biochemistry. They were the essential first stage that made all subsequent evolution possible. The beauty of this idea lies in the remarkable properties of lipids themselves. These molecules have a natural split personality. One end is hydrophilic, meaning it's attracted to water, while the other end is hydrophobic, repelled by it. When you have a collection of these molecules in water, they don't just float around randomly. To escape the water, their hydrophobic tails huddle together, while their water-loving heads face outward, spontaneously arranging themselves into perfectly sealed spheres called vesicles. These are incredibly simple prototypes of a cell membrane, and they form entirely on their own without any need for guidance or machinery. In the warm, nutrient-rich pools of the early Earth, such vesicles would have been forming and breaking apart constantly, a natural part of the environment. But the real magic begins when these simple lipid bubbles start to interact with the organic soup around them. As they form, they can easily trap whatever molecules are present in the water inside their hollow center. Imagine a primitive vesicle that just happens to encapsulate a few self-replicating RNA strands or a set of molecules that can perform a simple metabolic reaction. Suddenly, you have a tiny, isolated laboratory. The membrane protects these precious molecules, keeping them concentrated and close together so they can interact far more efficiently than they ever could out in the open ocean. This simple act of encapsulation creates the possibility for individuality and competition. One protocell might by chance contain a slightly better set of chemicals, allowing it to grow faster or become more stable than its neighbors. The most compelling evidence for this hypothesis comes from laboratory experiments that demonstrate just how lifelike these simple lipid vesicles can behave. Scientists have shown that under the right conditions, these vesicles can grow and even divide on their own without any complex biological instructions. For instance, if fresh lipid molecules are added to a solution containing vesicles, they can incorporate themselves into the existing membranes, causing the vesicles to grow larger. As they grow, they can become unstable and change shape, transforming from spheres into long, filamentous structures. With even the slightest external nudge, something as simple as a gentle water current, like waves lapping on a prehistoric shore, these filaments can pinch off into smaller, daughter vesicles, each containing a sample of the original internal mixture. This process of growth and division is a stunningly simple analog of cell reproduction, all driven by the basic physics and chemistry of the lipids themselves. It suggests that the fundamental cycle of life, growth and replication could have begun with these self-assembling structures, providing a stable platform upon which more complex genetic and metabolic systems could later evolve. Now, let's journey from the chemistry of ancient rocks and oily bubbles to a dimension that seems, at first glance, entirely incompatible with the warm, wet, messy world of biology, the bizarre and counterintuitive realm of quantum mechanics. We've explored theories grounded in geology and chemistry, but what if the spark of life required a set of rules from a completely different scale? This is the provocative and thrilling idea behind quantum biology, a field that suggests the strange phenomena of the quantum world, like tunneling, coherence, and entanglement, aren't just physics lab curiosities. They might be active, essential players in the fundamental processes of life, and they may have been crucial in guiding life's very first steps. For decades, biologists and physicists largely kept to their own domain. The general assumption was that the quantum world, with its spooky action at a distance and particles being in two places at once, was far too fragile to survive in the hot, noisy, and chaotic environment of a living cell. Quantum effects were thought to require near-absolute zero temperatures and perfect vacuums to be observed, not the warm soup of a bacterium. But a growing body of evidence is completely overturning that assumption. We're discovering that nature has somehow found a way to harness these quantum tricks, and the implications for life's origins are profound. It suggests that the universe's most fundamental laws might be written into the very blueprint of biology. One of the most well-documented examples of quantum mechanics at work in modern biology is a process called quantum tunneling. In the classical world we experience, if you roll a ball up a hill without enough energy to get over the top, it will roll back down. It's impossible for it to be on the other side unless it gets that extra push. But in the quantum world, particles like electrons don't play by those rules. Thanks to their wave-like nature, there's a small but real probability that they can simply appear on the other side of an energy barrier, as if they've dug a tunnel straight through the mountain. This isn't just theory, it's a cornerstone of modern physics, and it turns out our own bodies use it. Certain enzymes, the workhorse proteins that speed up biochemical reactions in every cell, appear to rely on quantum tunneling to do their jobs. Take the enzyme that helps manage our metabolism by transferring a hydrogen atom from one molecule to another. From a classical chemistry perspective, this reaction should be incredibly slow at body temperature. But it happens with astonishing speed. How research indicates the hydrogen atom, which is very light and therefore very quantum, doesn't climb over the energy barrier, it tunnels straight through it. This quantum effect makes the reaction trillions of times faster than it would be otherwise. Without quantum tunneling, basic processes that sustain life as we know it might simply not occur at a viable rate. So how does this connect to life's origins? If modern enzymes are harnessing quantum tunneling today, it's not a huge leap to propose that the very first primitive catalysts on the early Earth might have benefited from the same quantum assist. Perhaps the earliest self-replicating molecules or metabolic cycles were incredibly inefficient, operating at a pace so glacial that life would never have had time to emerge. Quantum tunneling could have provided a crucial shortcut, accelerating key reactions just enough to kickstart the whole process. In this view, the quantum world provided a hidden ladder that helped chemistry climb out of randomness and toward the organized complexity of life. But tunneling isn't the only quantum phenomenon that might have played a role. An even more mysterious and delicate effect, known as quantum coherence, might have been a master organizer in the primordial chaos. Coherence occurs when particles, like electrons, become linked and act as a unified wave, exploring multiple pathways simultaneously. This is famously put to use in photosynthesis. Plants, algae, and some bacteria need to transfer the energy they capture from sunlight to a reaction center where it can be stored. The problem is getting it there efficiently before it's lost as heat. Experiments have shown that this energy doesn't travel like a bouncing ball. Instead, it behaves like a wave of possibilities, spreading out and sampling all possible paths through the complex structure of the photosynthetic machinery at once. It essentially takes a quantum superhighway, finding the most efficient route in near-perfect efficiency before the delicate quantum state collapses. This isn't just a minor improvement, it's a dramatic enhancement of a process fundamental to most life on Earth. Applying this to life's origins opens up a breathtaking possibility. What if the same quantum coherence that allows a plant to harness sunlight today also helped organize the first energy harvesting systems billions of years ago? In the messy molecular soup before any complex structures existed, the transfer of energy from an abiotic source to a primitive metabolic cycle would have been wildly inefficient. But if groups of molecules could briefly exploit quantum coherence, they could have explored countless chemical configurations simultaneously, essentially conducting a massively parallel search for the most efficient way to capture and use energy. This quantum search function could have dramatically sped up the trial and error process of early evolution, guiding random chemistry toward the ordered, sustainable reactions that characterize life. It's a theory that suggests the universe, at its most fundamental level, might be biased toward creating complexity and order. But this exploration of quantum possibilities inevitably brings us back to Earth, to a more tangible and dramatic environment where chemistry and geology collide with raw power. We now venture from the subatomic realm to the steamy, bubbling, and mineral-rich landscapes of volcanic hot springs, a theory that paints a compelling picture of life's first stirrings happening not in the deep sea or a quiet pond, but on land in a dynamic and cyclic environment. This hypothesis suggests that the perfect cradle for life was a place of constant change, where wet and dry, hot and cool, organized and chaotic phases alternated in a rhythm that drove molecular complexity. The champion of this terrestrial idea is biochemist David Deamer, whose work has provided some of the most vivid experimental support for how life's building blocks could have been assembled. Deamer and others point to the numerous challenges of other origin sites. The deep ocean, while protected, is vast and dilute, making it difficult for sparse organic molecules to find each other and interact. The open ocean's primordial soup might have been too thin, its ingredients too scattered. Volcanic hot springs, on the other hand, offer a powerful solution to this concentration problem. Imagine a geothermal field on the early Earth, a landscape of porous rocks and clay surrounding pools of heated water, constantly fed by mineral-rich runoff and volcanic gases. This isn't a static environment, it's a natural laboratory with a built-in pump and cycle. The magic of this setting lies in its rhythm. During the day, the sun beats down and water from these shallow pools evaporates. As it does, it leaves behind everything that was dissolved in it, concentrating organic molecules onto the surfaces of clay and rock. This isn't a gentle process, it's a powerful focusing effect, taking a weak solution and turning it into a potent paste, forcing nucleotides, amino acids, and lipids into close proximity. Then at night or when rain falls, the dry crust is rehydrated. This cycle of wet and dry isn't just a simple repetition, it's an engine for chemistry. Each drying phase concentrates the reactants, and each wetting phase allows them to move and interact in new ways. Deamer's experiments have brilliantly demonstrated this. By simply taking simple fatty acids and nucleotides and subjecting them to repeated cycles of drying and rewarming in a simulated hot spring environment, his team has successfully driven the formation of RNA-like chains and, crucially, the encapsulation of these molecules within self-assembled lipid vesicles. This process creates protocells in a matter of days, not millennia, showing how a geothermal field could have been a prolific factory for the very structures we believe were life's precursors. Furthermore, these environments provide a rich and varied toolkit. The heat from the springs provides energy to drive reactions, while the mineral surfaces of clays and rocks act as catalysts, providing a solid support that organizes molecules and speeds up their assembly. The porous nature of the rock itself creates a multitude of micro-environments, tiny pockets and channels where different experiments in chemistry could be running simultaneously. One pocket might be perfect for forming lipids, while another nearby might be ideal for stringing nucleotides together. This diversity increases the chances that somewhere in one of these tiny rocky chambers, all the necessary components would eventually come together to form a self-sustaining, self-replicating system. It's a theory that combines the best aspects of many ideas: the catalytic surfaces of the clay world, the energetic drive of the iron-sulfur world, and the protective encapsulation of the lipid world, all orchestrated by the simple, powerful rhythm of evaporation and rain. While the theories we've explored so far have largely focused on Earth-bound energy sources from volcanic heat to chemical gradients, there exists another powerful force that permeates the entire cosmos, a relentless rain of high-energy particles that might have provided the critical spark for life's first ingredients. This brings us to a theory that looks beyond our planet's atmosphere to the violent hearts of distant supernovas, suggesting that cosmic radiation could have been the ultimate catalyst, jump-starting the chemistry of life not just here, but on countless worlds throughout the galaxy. It's a concept that turns a seemingly hostile aspect of space into a potential creative force. We often think of radiation as purely destructive, and for good reason. Cosmic rays, which are actually high-speed atomic nuclei propelled across the universe by exploding stars and black holes, carry immense energy. They can shred DNA and disrupt delicate electronics, posing a significant challenge for astronauts and long-term space travel. But on the early Earth, which lacked a protective ozone layer and a strong magnetic field, the planet was constantly bombarded by this intense radiation. Instead of being a barrier to life, this constant barrage might have been the very thing that provided the energy needed to forge complex molecules from simple ingredients. The same violent process that we shield ourselves from today could have been the universe's hammer and chisel, carving order from chaos. The experimental evidence for this is both compelling and elegant. Scientists have recreated the conditions of space and the early Earth's upper atmosphere in laboratories, using powerful radiation sources to simulate the effects of cosmic rays. In one famous series of experiments, researchers took a simple mixture of ice's water, methane, ammonia, and carbon monoxide, mimicking the composition of frozen interstellar dust grains or the icy surfaces of comets and moons. They then bombarded these ices with high-energy protons similar to those found in cosmic rays. The results were astonishing. This radiation-driven chemistry, rather than destroying the simple ices, transformed them. The energy from the radiation broke molecular bonds, but in doing so, it created highly reactive fragments that quickly recombined into more complex structures. Analysis revealed that this process produced a rich array of organic compounds, including numerous amino acids, the very building blocks of proteins. This suggests a profoundly beautiful and universal mechanism. Icy comets, asteroids, and the dense clouds of gas and dust between stars are constantly being zapped by cosmic rays. Each impact acts like a tiny, intense chemistry set, driving reactions that would be impossibly slow under cold, dark conditions. Over millions of years, these cosmic laboratories could have manufactured vast quantities of life's precursor molecules. Then, through impacts and gravitational nudges, these pre-processed organic-rich bodies could have been delivered to young planets like Earth, essentially seeding them with a pre-made starter kit for life. This theory beautifully bridges the gap between panspermia and terrestrial origin theories. It's not that life itself traveled here, but the activated, complex chemistry that makes life possible was manufactured in the depths of space and delivered ready for assembly. It paints a picture of a galaxy that is not only habitable but actively productive, where the most destructive forces in the universe are also, paradoxically, among the most creative. This brings us to a fascinating synthesis that elegantly bridges the deep ocean and the volcanic highlands. It's a theory that argues the perfect cradle for life wasn't found in one single, stable environment, but in a place of constant dynamic change, a zone where the chemistries of land and sea intertwine. Welcome to the hydrothermal field theory, a compelling model that proposes life emerged in the vibrant, fluctuating landscapes of coastal geothermal fields, where the very instability of the conditions provided the creative engine for complexity. Imagine the shoreline of the early Earth, a place of raw, untamed power. Here, volcanic activity heats the ground, creating not just isolated hot springs, but vast, interconnected fields of geysers, bubbling mud pots, and steaming pools. Crucially, these aren't sealed-off systems. They exist at the boundary between land and sea. Tidal flows constantly wash ocean water through these porous, mineral-rich rocks, while rainwater and evaporation add their own layers to the mix. This creates an environment that is never static. The temperature, the pH, the salinity, and the concentration of dissolved minerals in each pool are in a perpetual state of flux, shifting with the daily tides, the heat from below, and the rhythms of sun and rain. Where other theories might see a chaotic, hostile mess, this theory sees a perfect laboratory. It's the difference between trying to cook a complex meal over a single, steady flame versus having a full kitchen with a simmering pot, a hot oven, a blender, and a cold fridge all working in concert. Each change in condition drives a different kind of chemistry, pushing molecules to assemble, disassemble, and reconfigure in new ways. The genius of this model is how it solves the problem of dilution and concentration that plagues both the open ocean soup and the deep sea vent hypotheses. In a vast ocean, molecules are too spread out to interact meaningfully. At a deep sea vent, they might be concentrated, but they are also at risk of being washed away by the constant, powerful flow of vent fluid. A coastal hydrothermal field offers the best of both worlds. The porous volcanic rock acts like a massive, natural sponge. As heated water rises from below and cool seawater percolates down from above, this sponge soaks up organic molecules, trapping them within a maze of microscopic pores and channels. This isn't a passive filter, it's an active reactor. Each tiny pore becomes its own isolated micro-laboratory, safely concentrating a unique mix of ingredients away from the diluting effect of the open water. Now add the power of cycles. The theory posits that it was this relentless rhythm of change, this pumping action of the environment itself, that was the true driver of life's emergence. During low tide or under a hot sun, a pool might partially evaporate, concentrating its trapped molecules to a thick paste and even driving the formation of longer molecular chains, just as David Deemer's experiments showed. Then the tide returns or a rain shower falls, suddenly rehydrating the system. This influx of cooler, chemically different water doesn't just dilute the mixture, it introduces new ingredients and creates new energy gradients, sparking a fresh round of reactions. This cycle of wet and dry, hot and cool, acidic and alkaline would have acted like a natural PCR machine, endlessly copying, selecting, and amplifying the most stable and efficient molecular structures. A lipid that can form a more resilient vesicle or an RNA strand that can slightly aid a reaction would have a distinct advantage. With each cycle, these winners would become more abundant, slowly inching toward the threshold of a living system. It's a vision of life not as a sudden spark in a stable crucible, but as a melody that slowly emerged from the rhythm of the planet itself. While we often look to the dramatic forces of our own planet for life's origins, we must remember that Earth has never been an isolated system. It has been constantly bombarded by visitors from space since its very formation, and some of these celestial wanderers may have carried with them the most precious cargo imaginable, the pre-formed ingredients for life itself. This leads us to a theory that is less about how life began and more about how its essential building blocks were delivered, pre-packaged to a waiting world. It's the concept of meteorite delivery, and it's supported by some of the most tangible and extraordinary evidence we have. The key players in this cosmic delivery service are a specific class of space rocks known as carbonaceous chondrites. These are among the most primitive materials in our solar system, ancient relics that have remained largely unchanged since its formation over 4.5 billion years ago. Unlike the metallic or rocky meteorites that often make the news, carbonaceous chondrites are dark, crumbly, and packed with carbon and water. They are essentially cosmic time capsules, preserving a snapshot of the chemistry that was present in the dusty disk that swirled around our infant sun. And within that chemistry, scientists have found a treasure trove of organic compounds that would have been absolutely vital for kickstarting life on any planet they landed on. To understand just how rich these meteorites are, we need look no further than one of the most studied rocks in the history of science, the Murchison meteorite. This particular chunk of space debris fell to Earth in a fireball over Murchison, Australia, in September of 1969, scattering fragments across the countryside. Its timing was perfect as the Apollo moon missions were bringing back pristine extraterrestrial samples and scientific techniques were advancing rapidly. When researchers began to analyze the Murchison meteorite, they were utterly astonished. It wasn't just a simple rock, it was a complex chemical factory. The Murchison meteorite was found to contain a stunning array of organic molecules. Most importantly, it wasn't just simple carbon chains. Analysis revealed the presence of over 90 different amino acids, the fundamental building blocks of proteins. What's truly mind-bending is that about half of these amino acids are common here on Earth and are used by all known life forms. The other half, however, are rare or entirely non-existent in terrestrial biology. This crucial detail proves that these compounds were formed by non-biological abiotic processes in space, ruling out the possibility that the meteorite was merely contaminated after it landed. But the inventory doesn't stop there. The Murchison meteorite also contains nucleobases, which are the information-carrying components of DNA and RNA, such as uracil and xanthine. Even more remarkably, recent analyses have confirmed the presence of sugars, including ribose, the essential R in RNA, the very backbone of the molecule that might have been the first genetic material. This isn't an isolated case. Other carbonaceous chondrites, like the one that fell near Tagish Lake in Canada in 2000, have shown similar complex organic profiles. The implication is staggering. Billions of years ago, during the period known as the Late Heavy Bombardment, Earth was under a constant and intense barrage from asteroids and comets. This was a violent and chaotic time, but it may also have been a period of immense fertilization. Each impact from a carbonaceous chondrite would have been like a targeted supply drop, scattering its payload of amino acids, nucleobases, and sugars across the entire planet. These organics would have rained down into the early oceans, tidal pools, and hot springs, dramatically enriching the primordial soup and providing a concentrated, pre-made toolkit for the chemistry of life to begin. This theory doesn't necessarily compete with ideas like the RNA world or hydrothermal vents. Instead, it complements them perfectly. It suggests that while Earth provided the welcoming environment and the necessary energy, the universe might have provided a critical head start by delivering a significant portion of the raw materials, pre-synthesized in the vast chemical laboratories of interstellar space and the early solar system. But this intricate dance of molecules, whether organized by mineral surfaces or concentrated in tidal pools, still faces a fundamental hurdle. How could a system of chemical reactions become self-sustaining, able to not just create complexity but also maintain and perpetuate itself? This brings us to a truly fascinating and profound theory that shifts the focus from individual molecules to the power of the network itself. It's the concept of autocatalytic sets, a model that suggests life's first glimmer wasn't a single replicator, but an entire web of chemical reactions that collectively bootstrapped themselves into existence, creating a primitive form of metabolism long before genetics ever entered the picture. To grasp this elegant idea, we need to understand the word autocatalytic. A catalyst is something that speeds up a chemical reaction without being consumed by it. An autocatalyst is a reaction product that catalyzes its own formation. A simple, well-known example is the formation of hydrocyanic acid. Under the right conditions, hydrocyanic acid molecules can catalyze the formation of more hydrocyanic acid from its components. Now, imagine scaling this idea up dramatically. An autocatalytic set is a closed network of reactions where every single product in the web is created by a reaction that is itself catalyzed by another member of the set. It's a self-fueling, self-sustaining chemical cycle, a closed loop of creation where the whole network maintains and reproduces itself. In this view, the first living system wasn't a thing but a process, not a molecule but a dynamic, sustainable pattern of chemical flow. This revolutionary framework was most powerfully developed by the theoretical biologist and complex systems researcher Stuart Kauffman, starting in the 1970s. Kauffman used sophisticated mathematics and computer modeling to demonstrate something astonishingly simple yet profound. As a random chemical soup grows more complex, reaching a critical diversity of molecules, the chance formation of an autocatalytic set doesn't just become possible, it becomes virtually inevitable. It's a phase transition in chemistry, much like water spontaneously freezing into ice once it hits a certain temperature. Once you have enough different kinds of molecules bumping into each other, the law of large numbers dictates that some will inevitably begin to catalyze the reactions that form others, stitching themselves together into a self-sustaining web. Kauffman's genius was in showing that this emergence of order isn't a miraculous accident. It's a natural consequence of complexity, an expected property of the chemical universe waiting to express itself under the right conditions. The beauty of this theory is how it completely reframes the origin of life problem. Instead of the incredibly unlikely event of a single, perfect self-replicating molecule like RNA assembling by chance, life could have begun as a collective effort. A ragtag group of simple molecules, none of which can replicate on their own, could have achieved a kind of chemical democracy where each member helps create another. One molecule might catalyze the formation of a second, which in turn catalyzes the formation of a third, and that third one might close the loop by catalyzing the reformation of the first. This system wouldn't have a genome or a blueprint. Its information would be stored in the stable, self-perpetuating structure of the network itself. This collective autocatalysis represents a primitive form of metabolism, a system that can harness energy from its environment to maintain its own organized state far from chemical equilibrium, which is a key hallmark of life. Experimental evidence for this has moved from mathematical elegance to tangible reality in the lab. Renowned origin of life researcher Gunther von Kiedrauski demonstrated a simple autocatalytic cycle with synthetic DNA fragments in the 1990s. More recently, the team of brilliant chemist Lee Cronin at the University of Glasgow has shown something even more striking. They've created what they call inorganic chemical cells, or iChel. By mixing simple metal oxides, common, abundant minerals, in a solution, they observed the spontaneous formation of a network of reactions where the products catalyze the creation of more cells. These are not alive, but they exhibit lifelike behaviors. They grow, they divide, and they maintain a stable internal environment. They are, in essence, a working model of a simple autocatalytic set, showing that the spontaneous emergence of self-sustaining chemical networks is not just a theoretical possibility, but a demonstrable chemical reality. This suggests that the path to life may have been paved not by a single golden molecule, but by the collective power of chemistry finding its own stable, self-reinforcing rhythms. While the warmth of hydrothermal vents and the energy of hot springs offer compelling narratives, there's a chillingly beautiful alternative that suggests life's first steps may have required not heat but cold, a deep freeze that provided protection, concentration, and the gift of time. This is the theory that life began not in a warm little pond, but in a cold little ice pocket where the seemingly hostile environment of ice acted as a natural laboratory, preserving delicate chemistry and allowing for reactions of incredible precision. We often think of ice as a barren, static state, but in reality, It's a dynamic and surprisingly complex environment, especially when we consider the microscopic world within it. When water freezes, it doesn't form a perfect solid block. Instead, it creates an intricate lattice, and as it does, it pushes out impurities, salts, gases, and any organic molecules that were dissolved in it. These impurities don't just vanish, they become concentrated into tiny, microscopic pockets of liquid brine trapped within the solid ice matrix. These briny microenvironments are incredibly rich soups with the dissolved molecules forced into close proximity. This solves the critical problem of dilution that plagues many ocean-based origin theories. The ice actively concentrates the ingredients for life, creating billions of tiny, natural test tubes all working in parallel. Within these icy pockets, the cold itself becomes a major advantage. Chemical reactions, generally speaking, slow down at lower temperatures. While this might seem like a drawback for the fragile, nascent molecules of early life, it was a lifesaver. The intense ultraviolet radiation from the young sun would have been devastating to complex organics on the surface or in shallow waters. But a layer of ice, even a relatively thin one, acts as a superb shield, filtering out this destructive UV light and providing a safe haven for chemistry to proceed without constant interruption. Furthermore, the slowed reaction rates in the cold are not always a bad thing. They allow for a level of control and precision that would be impossible in a hot, chaotic environment. Unwanted side reactions that would quickly destroy a delicate molecule at high temperatures are dramatically reduced. This allows for more selective and orderly bond formation, giving complex structures like RNA a much better chance to assemble correctly and survive long enough to become functional. The most compelling modern evidence for this icy cradle theory doesn't just come from lab experiments, though those are powerful, but from our robotic explorations of the outer solar system. We now have stunning examples of icy worlds that are considered among the most promising places to find extraterrestrial life today: Jupiter's moon Europa and Saturn's moon Enceladus. Both are frozen spheres, but beneath their icy crusts, we have strong evidence for vast, global liquid water oceans, kept warm by the gravitational flexing of their host planets. These are not dead, frozen wastelands. They are dynamic worlds with energy sources, liquid water, and all the ingredients we believe are necessary for life. They serve as a powerful proof of concept that ice is not a tomb, but a protective shell for potentially thriving aquatic environments. The plumes of water vapor and organic molecules erupting from Enceladus's south pole are a direct sample of that hidden ocean, telling us that the chemistry of life can not only survive but potentially flourish in such a setting. Back on Earth, scientists have put the ice theory to the test in the laboratory with remarkable results. Researchers have taken simple mixtures of water, ammonia, and cyanide, common compounds thought to be present on the early Earth and in space, and frozen them solid. They then expose these icy mixtures to ultraviolet radiation, simulating the sun's light. The results are extraordinary. This process doesn't just preserve the molecules, it actively builds them. The energy from the radiation drives reactions within the ice, leading to the formation of a plethora of organic compounds, including nucleobases like uracil and cytosine, which are vital components of RNA. The ice matrix itself plays a crucial role, holding the molecules in a fixed orientation that facilitates these specific reactions, acting as both a reactor and a catalyst. This demonstrates that the cold of space or a frozen early Earth wasn't a barrier to life's chemistry. It was an essential facilitator, providing the perfect conditions to manufacture and safeguard the very building blocks of biology. Now we descend from icy space and sunlit hot springs into the very bedrock of our planet, to a realm of immense pressure and perpetual darkness that challenges our very definition of a habitable world. This is the theory of the deep Earth biosphere, a hypothesis proposing that life may not have begun on the planet's dynamic surface, but was instead born and nurtured in the stable, energy-rich depths of the Earth's crust, entirely isolated from the sun-driven world above. It suggests our search for life's origins has been looking in the wrong place all along, and that the cradle of life was not a pond or a vent, but a tiny, pressurized pore within a rock miles underground. The conditions there are almost unimaginably extreme. We are talking about an environment of total blackness, with crushing pressures hundreds of times greater than at the surface, and temperatures that can soar well past the boiling point of water. There is no oxygen, no sunlight, and barely any free water. To our surface-dwelling minds, this seems like the most hostile and sterile environment possible. Yet, astoundingly, this is not the case. Over the past few decades, through ambitious drilling projects that have plumbed the continental crust and the ocean floor, scientists have uncovered a vast, hidden ecosystem thriving in this subterranean prison. We're not talking about a few scattered, hardy survivors. We're talking about a massive, diverse biosphere whose total mass might rival or even exceed all life on the surface. This discovery of a dark life completely independent of the sun forces us to rewrite the textbooks on biology and consider the possibility that the surface world, for all its beauty, might just be the tip of the iceberg when it comes to life on Earth. The inhabitants of this deep biosphere are almost exclusively microbes, primarily bacteria and archaea. These are not the familiar germs we know. They are extremophiles of the highest order, and they have invented entirely different ways to live. Without sunlight, they cannot perform photosynthesis. Instead, they are chemolithiotrophs, a complex word that breaks down into a simple, brilliant concept: they eat rocks. These microbes derive their energy not from organic matter or the sun, but from the fundamental geochemistry of the planet itself. They catalyze reactions between inorganic minerals and gases dissolved in water that percolate slowly through the bedrock. For example, some microbes can oxidize hydrogen gas that is released when water reacts with iron-rich minerals like olivine. Others can reduce carbon dioxide or sulfate, using the energy released from these reactions to power their metabolisms and build their cellular structures. Their entire existence is fueled by the slow, steady exhale of the planet's geothermal energy and the radioactive decay of elements within the rock. This is a completely self-contained world, a closed loop of chemical energy that has been running for billions of years, utterly oblivious to day and night, summer and winter. This profound discovery lends incredible weight to the idea that life could have originated in such a setting. The deep subsurface offers some unique advantages that the chaotic surface world lacked billions of years ago. Most importantly, it provides extraordinary protection. During Earth's early history, the surface was a shooting gallery, bombarded by asteroids and comets in the late heavy bombardment. These impacts would have been sterilizing events, vaporizing the oceans and scorching the land, likely wiping out any nascent life forms trying to establish a foothold. But deep underground, several miles beneath the surface, life would have been insulated from this cosmic violence. The temperature would have remained stable, and the intense ultraviolet radiation from the young, violent sun would have been completely blocked. The deep crust would have been a safe bunker, a constant and stable refuge where delicate chemical processes could proceed for millions of years without interruption. Furthermore, the environment provides a perfect natural laboratory for the chemistry of life. The porous, fractured rock acts like a massive, three-dimensional sponge, creating billions of microscopic chambers and channels. Within these tiny pores, simple organic molecules formed from geothermal gases or delivered by percolating water could become highly concentrated, dramatically increasing the chances they would interact and form more complex structures. The extensive mineral surfaces, rich in reactive metals like iron, nickel, and sulfur, could have acted as the first catalysts, organizing molecules and facilitating the key reactions that led to self-replication and metabolism. In many ways, the theory of a deep biosphere origin synthesizes other ideas we've discussed. It has the catalytic mineral surfaces of the iron-sulfur world, the protected, concentrated environment of the hydrothermal field theory, and the energy-rich, chemically reducing conditions that would have been ideal for forging the first biological molecules. It presents a compelling vision of life as an inherent property of a geologically active rocky planet, emerging from the very chemistry of its interior, safe and secure in the planet's deep embrace, waiting for the surface to become calm enough to eventually emerge and bloom. While the deep Earth offers a sheltered refuge, another theory proposes that life's first spark required the exact opposite, a direct and powerful blast of raw energy from the sky. This idea suggests that the very thing we now shield ourselves from, the intense, unfiltered ultraviolet radiation from a young, violent sun, was the essential catalyst that cooked the first ingredients of life in the atmosphere itself. This is the theory of photochemical origins, and it paints a vivid picture of our planet's upper air transforming into a vast cosmic chemistry set, with each photon of light acting as a precise tool to forge complexity from simple gases. To understand why this would work, we have to reimagine Earth's earliest atmosphere. Billions of years ago, before plants filled the air with oxygen, our sky was a very different place. It was a thick haze of what chemists call reducing gases, things like methane, ammonia, water vapor, and carbon monoxide. Crucially, there was no ozone layer. This meant that the planet was utterly exposed to the full, ferocious intensity of ultraviolet radiation streaming from the sun. From our modern perspective, this sounds like a nightmare scenario, a constant sterilizing beam scouring the surface. But for probiotic chemistry, this UV light was not a destroyer. It was the ultimate power source. Each photon carried enough energy to break the strong chemical bonds holding these simple gas molecules together, shattering them into highly reactive fragments known as free radicals. It's in the frantic dance of these free radicals that the magic happened. These fragments, desperate to regain stability, would collide and recombine with other broken pieces and intact molecules in a frenzied game of chemical musical chairs. And through this chaotic process, driven entirely by the sun's energy, more complex organic molecules began to form. One of the most important products of this atmospheric photochemistry was formaldehyde. This simple molecule, which we know today as a preservative, is a crucial biochemical building block. It's incredibly reactive and can easily link up with other molecules to form sugars, including ribose, the essential sugar backbone of RNA. So high in the ancient sky, ultraviolet light was essentially manufacturing the fundamental components of genetics and metabolism, raining them down onto the waiting surface below. This isn't just a theoretical idea. It's been demonstrated in spectacular laboratory experiments that recreate these ancient conditions. Scientists construct glass chambers filled with the presumed primordial gas mixture, methane, ammonia, water vapor, and then subject it to a powerful UV lamp simulating the young sun. Within hours, the clear glass becomes clouded with a faint brownish haze. This haze, when analyzed, is found to be teeming with organic compounds, a complex tarry substance known as tholins. And within this synthetic primordial goo, researchers find a rich array of molecules critical for life: amino acids, the building blocks of proteins, and nucleobases, the information-carrying letters of DNA and RNA. These experiments provide tangible proof that the simple act of shining light on basic gases can generate the sophisticated chemistry that life is built from, turning the entire atmosphere into a prolific factory for life's raw materials. This journey through the extremes of pressure and radiation brings us to a theory that combines the shelter of the deep Earth with the creative chaos of space, proposing that the perfect nursery for life wasn't a planet at all, but a celestial wanderer. We've considered how life's building blocks were delivered by meteorites, but what if those building blocks were not just assembled, but actually sparked into existence aboard the delivery vehicle itself? This is the theory of cometary nurseries, a thrilling extension of panspermia that suggests the very first living organisms may have awakened in the warm, watery interiors of comets long before they ever reached a planet like Earth. When we picture a comet, we typically imagine a dirty snowball, a frozen relic from the solar system's birth hurtling through the unimaginable cold of space. It seems like the least likely place for life to begin, but this classic view is being radically overhauled by new discoveries. We now understand that comets are not simple, inert lumps of ice. They are complex, dynamic mini-worlds, and their interiors can be surprisingly active. The key is radioactivity. Comets, especially larger ones, contain radioactive isotopes of elements like aluminum and potassium that were incorporated during their formation. As these isotopes decay, they release heat. In the core of a sufficiently large comet, this radioactive heating can be significant enough to melt the primordial ice, creating vast, hidden reservoirs of liquid water. Imagine the interior of one of these ancient, kilometer-wide comets in the early solar system. Deep within its frozen shell, a secret ocean is kept liquid by nuclear decay. This subsurface sea is not a calm, placid lake. It's a turbulent environment, constantly churned by the heat from within and the tidal forces from the comet's rotation and any nearby gravitational pulls. And this water is anything but pure. It is a rich, dense broth, saturated with all the complex organic compounds that we know form on the surfaces of interstellar dust grains, and within the ice matrix itself, amino acids, nucleobases, sugars, and lipids. The comet's interior becomes a natural pressure cooker, concentrating all the ingredients for life into a protected, liquid environment, shielded from the harsh radiation of space by layers of overlying rock and ice. This setting provides a stunningly complete package for life's origins, combining the benefits of several other theories we've explored. It offers the concentration of ingredients that solves the dilution problem of the open ocean. It provides the protected, stable environment of the deep Earth biosphere, safe from the sterilizing impacts that ravaged the early planetary surfaces. The porous structure of the comet's interior, full of cracks and chambers in the rock and ice, offers countless mineral surfaces that could act as catalysts, much like the clays or pyrite of other theories. And the constant churning and temperature gradients within this watery pocket could drive the same kind of cyclic processes that make hydrothermal fields so compelling, encouraging molecules to link up, encapsulate, and evolve. In this view, a comet is not merely a delivery truck. It is a fully equipped mobile laboratory, cruising through the solar system and actively conducting the experiment of life within itself. The most tantalizing evidence for this idea comes from our own robotic explorations. The European Space Agency's Rosetta mission, which spent years studying comet 67P/Churyumov-Gerasimenko, found a landscape far more complex and varied than anyone had predicted. The probe discovered a wealth of organic molecules on the comet's surface, including the amino acid glycine and the key element phosphorus, both essential for life as we know it. While 67P is likely too small to host liquid water today, its chemical complexity strongly suggests that the ingredients were all there. For larger comets in the early, warmer solar system, the conditions would have been even more ideal. This theory elegantly bridges the gap between panspermia and terrestrial origin stories. It suggests that life may not have begun on a planet, but that planets were the fertile gardens where cometary seeds, already pre-germinated, finally found the stable conditions to truly take root and flourish across the cosmos. But if RNA, for all its elegant versatility, still presents a formidable challenge for prebiotic synthesis, perhaps there was an even simpler molecule that paved the way. We now turn to a theory that asks a deceptively simple question. What if the very first genetic system wasn't based on a molecule we find in our cells today, but on a sturdier, more straightforward cousin that has since been made obsolete? This is the intriguing hypothesis of a peptide nucleic acid world, a concept that proposes a bridge between the world of proteins and the world of genetics long before either had fully emerged. To understand why PNA is such a compelling candidate, we need to look at its unique structure. The peptide in its name is the key. In modern cells, the backbone of DNA and RNA is made of sugar and phosphate groups, which are complex molecules that can be difficult to form and are notoriously unstable, especially in the heat and variable pH of the early Earth. PNA replaces that fragile sugar-phosphate backbone with something far more robust, a chain of amino acids linked by sturdy peptide bonds, identical to the bonds that hold proteins together. This simple substitution creates a molecule of remarkable power. Its backbone is neutral, lacking the negative electrical charge that makes RNA and DNA repel each other and require complex helpers to interact. This neutral spine allows PNA strands to bind to each other and to DNA and RNA with even greater strength and specificity than the natural molecules can achieve on their own. It's as if nature's first attempt at a genetic system used a simple, durable nylon rope instead of the more delicate, intricate chain it eventually settled on. This structural simplicity solves two of the biggest headaches in the RNA world hypothesis. First, the building blocks of PNA's backbone. Simple amino acids like glycine or alanine were almost certainly abundant on the early Earth, far easier to form abiotically than RNA complex ribose sugar. Second, PNA is incredibly stable. It isn't easily broken down by water or heat, meaning it could have persisted and accumulated in the chaotic environments of tidal pools or hydrothermal vents, surviving long enough to undergo the random interactions that could lead to self-replication. Its talent for base pairing is its masterstroke. Just like DNA and RNA, PNA uses the same four nucleobases adenine, thymine, cytosine, and guanine to store information. A PNA strand can form a classic double helix with a complementary strand of PNA, or even with DNA or RNA, following the same base pairing rules that are the universal language of genetics. This means a PNA world could have seamlessly handed off its genetic information to a nascent RNA world, acting as a stable training ground where the principles of Darwinian evolution were first learned using a simpler, more forgiving molecule. The most captivating implication of the PNA world theory is how it elegantly bridges the great divide between the two fundamental pillars of biology, genetics and metabolism. For decades, origin of life research has been split into genetics first and metabolism first camps. PNA offers a beautiful synthesis. Its very structure is a hybrid. It is a genetic molecule built from the same stuff as proteins. This suggests that the dichotomy might be a false one. Perhaps the first replicators weren't purely informational or purely metabolic, but something in between. A world based on PNA could have seen its first replicating molecules also performing rudimentary catalytic functions, blurring the line between gene and enzyme from the very beginning. This molecule, born from the simple marriage of abundant amino acids and nucleobases, could have been the universal pioneer, sturdy enough to establish the rules of the game and stable enough to hold the door open for its more sophisticated, but more fragile, eventual successor. While we often picture life's origins in terms of chemistry, energy, and protection, there's an invisible force that blankets our planet, one that might have played a far more active role in guiding life's first steps than we ever imagined. This force is Earth's magnetic field, a powerful shield that deflects the solar wind and cosmic radiation. But beyond its protective role, this field might have done something even more profound. It could have provided a subtle directional guide, a kind of cosmic choreography that helped organize the chaotic dance of molecules toward the first rhythms of life. This theory suggests that the planet itself, through its magnetic heartbeat, lent a gentle hand to the process of biogenesis. To understand how, we need to think about the nature of the early Earth's magnetic field and the building blocks it might have influenced. Billions of years ago, the field was likely even stronger than it is today, generated by the furious churning of the planet's molten iron core. This creates a vast, invisible architecture of magnetic field lines that arc from pole to pole, creating a directional force that permeates everything. Now consider the molecules we've been discussing. Many of them, especially in the charged environments near hydrothermal vents or in evaporating tidal pools, would have carried an electrical charge. Amino acids, nucleotides, and even the lipids that form vesicles can act as tiny, floating magnets, orienting themselves along these planetary field lines. This alignment isn't a powerful force, but on the molecular scale, even a gentle nudge can be enough to make a critical difference. The most significant potential benefit of this magnetic guidance would have been in the incredibly difficult process of molecular self-assembly, particularly for something like the first nucleic acids. For one RNA strand to act as a template to copy itself, the free-floating nucleotides need to line up in exactly the right order and orientation against it. In a chaotic, jostling soup, this is like trying to complete a complex puzzle during an earthquake. The magnetic field could have provided a steadying influence. By gently encouraging these charged molecules to align along a north-south axis, the field would have dramatically increased the odds that they would bump into their template in the correct way, essentially increasing the efficiency of these first clumsy replication attempts. It's a fascinating idea that the planet's magnetic field acted as a silent, universal lab assistant, holding the test tube steady while the most delicate work was being done. This isn't just speculative physics. We have a stunning piece of modern biological evidence that suggests ancient life not only experienced this force but learned to harness it. Meet the magnetotactic bacteria. These modern microorganisms are living fossils, possessing a biological feature that seems ripped right from this theory. They fabricate their own perfect microscopic crystals of magnetic minerals like magnetite or graphite, arranging them into a tiny internal compass needle called a magnetosome. Using this built-in guidance system, these bacteria actively swim along the Earth's magnetic field lines. In the northern hemisphere, they use it to guide them downward, away from the oxygen-rich surface water that is toxic to them, and toward the safer, low-oxygen sediments they prefer. It's a sophisticated navigation system that has been perfected by evolution. The existence of magnetotactic bacteria is powerful because it implies a very deep and ancient relationship between biology and geomagnetism. This intricate ability to biomineralize perfect magnetic crystals and integrate them into a sensory apparatus didn't appear overnight. It suggests an evolutionary history stretching back to a time when the magnetic field was a much more dominant environmental factor, perhaps even a tool that the very first life forms learned to use. These modern bacteria could be the living relics of a far more ancient time, a time when the magnetic field wasn't just a tool for navigation, but was fundamentally intertwined with the very organization and replication of the earliest living systems. They are a clue that the invisible force that protects our planet may have also helped to create the life it now shields. Now, let's shift our focus from the cosmic and the quantum to a setting much closer to home, one that you might have actually explored yourself on a rocky beach, the humble tidal pool. This theory suggests that the secret to life's assembly wasn't a singular, dramatic event, but a gentle, relentless rhythm. The patient, pulsing beat of the tides, which provided the perfect natural machinery to concentrate life's scattered ingredients and patiently forge them into something new. This is the idea of tidal pool concentration, a beautifully simple yet powerful concept that leverages one of the most fundamental cycles on our planet. Imagine the shoreline of the early Earth, constantly reshaped by the gravitational pull of a closer, younger moon. In this dynamic zone, shallow depressions in the volcanic rock would fill with ocean water at high tide, creating temporary pools rich with the organic molecules we've discussed, those formed in the atmosphere, delivered by meteorites or washed in from the sea. As the tide recedes and the sun beats down, these isolated pools begin to evaporate. This isn't a gentle process, it's a powerful act of concentration. The water vanishes, but every single amino acid, nucleotide, and lipid that was dissolved in it is left behind. What was once a weak, diluted soup becomes an intensely potent paste, forcing these molecules into incredibly close proximity on the surfaces of warm rocks and clays. They are no longer lonely entities drifting in a vast ocean. They are neighbors pressed together, compelled to interact. Then the cycle reverses. The tide returns and cool, fresh seawater surges back in, rapidly rehydrating the dried, concentrated film. This sudden influx doesn't just dilute the mixture again. It introduces a shock of new energy and new ingredients, while also allowing the now concentrated molecules to move and collide with incredible force. This continuous loop of wet and dry, of concentration and reaction, is like a natural planetary scale PCR machine. Each drying phase is a focusing step gathering the raw materials. Each wetting phase is a mixing step allowing new combinations to form. With every single tide over millions of years, this process would have relentlessly driven chemistry forward, dramatically increasing the odds that random interactions would lead to something meaningful. The true genius of this mechanism lies in how it solves one of the most stubborn problems in prebiotic chemistry: how to form long polymers. Linking individual nucleotides into a chain of RNA or amino acids into a protein is a dehydration reaction. It requires the removal of a water molecule to form a new bond. In a vast, watery ocean, this is incredibly difficult. The law of mass action favors the reverse reaction, hydrolysis, which breaks bonds apart. But in an evaporating tidal pool, this problem vanishes. The environment itself is actively removing water, physically driving the chemistry of polymerization. As the pool dries, molecules are not just concentrated, they are being pushed into forming the very bonds that would eventually become the backbone of genetics and metabolism. Laboratory experiments have powerfully confirmed this. Researchers have shown that simply subjecting solutions of amino acids or nucleotides to repeated wet-dry cycles, just like those in a tidal pool, readily drives the formation of protein-like chains and RNA-like polymers, all without the need for complex enzymes or catalysts. It's a stunning demonstration of how the patient, rhythmic pulse of our planet could have provided the precise conditions needed to assemble the first fragile architectures of life. Yet for all the dynamic environments we've explored, from the churning oceans to the pulsing tidal pools, there's a theory that suggests the most critical work might have been done on the most miniature of stages, not here on Earth, but suspended in the silent, vast emptiness between the stars. This is the concept of cosmic dust grain catalysis, a beautifully elegant idea that proposes the very first steps toward life's complexity didn't require a planet at all, but instead took place on the surfaces of tiny, floating motes of dust that permeate the galaxy. To understand how this could work, we need to reimagine the challenge of building complex molecules in space. In the near-perfect vacuum of an interstellar cloud, atoms and simple molecules are incredibly sparse. For two of them to meet, react, and form a bond is a fantastically unlikely event. They are simply too far apart, but interstellar space is not entirely empty. It's filled with a fine mist of microscopic dust grains, often just a few hundred atoms across, made of silicates or carbon, similar to soot or sand. These tiny particles act as miraculous meeting points. When a lone hydrogen atom or a simple carbon monoxide molecule drifting through the void bumps into one of these dust grains, it doesn't just bounce off. At the frigid temperatures of space, around -260 degrees Celsius, it sticks to the surface through a weak electrical force known as the van der Waals force. Over immense stretches of time, these grains become coated in a frosty mantle of frozen ices and simple atoms, all held in place and, most importantly, held in close proximity to one another. This is where the magic happens. While these atoms are trapped on the grain's surface, they aren't completely immobile. They can still skitter and diffuse across this frozen landscape. Suddenly, the impossible becomes probable. Two atoms that would have taken millennia to find each other in the open void are now next-door neighbors on a cosmic speck of dust. When they finally meet, they can react, forming a new, more complex molecule that itself becomes part of the icy coating. This process allows for a gradual build-up of molecular complexity, one reaction at a time, all facilitated by the simple presence of a solid surface. These dust grains become the universe's smallest and most prolific chemistry labs, silently constructing the ingredients of life in the dark clouds between the stars. Perhaps the most profound mystery this theory helps to solve is the origin of life's handedness or chirality. Many of the molecules essential for life, like amino acids, come in two mirror image forms, much like your left and right hands. They are chemically identical, but their structures are mirror opposites. In a lab, any abiotic reaction that creates amino acids will produce a perfect 50-50 mixture of both left and right-handed versions. Yet, every single known life form on Earth uses only left-handed amino acids to build its proteins and only right-handed sugars in its DNA backbone. This uniform homochirality isn't just a quirky detail, it's absolutely essential. For life's machinery to work, for enzymes to fit onto their targets, and for the double helix to form properly, every single piece must have the same handedness. A mixed-handed system would be a dysfunctional mess, like trying to assemble a machine using both left-handed and right-handed screws. This is where cosmic dust grains offer a stunningly elegant solution. These grains aren't just passive platforms. Many of them are likely asymmetric themselves, with surfaces that have a slight inherent handedness due to their crystalline structure. Furthermore, they are constantly bathed in circularly polarized light, a special kind of starlight that spirals through space like a corkscrew, either clockwise or counterclockwise. This light can selectively break down one mirror image form of a molecule slightly more than the other. The theory suggests that on the surface of a dust grain, these effects could work in concert. The grain's surface might provide a template that favors the formation of one-handedness, while the polarized light selectively destroys the other. Over millions of years, this could result in a tiny grain of dust whose icy mantle is subtly enriched with, say, left-handed amino acids. When such grains eventually clump together to form comets and asteroids, and those bodies later crash into young planets like Earth, they would deliver this pre-bias, this slight excess of one-handedness, seeding the planet with the foundational asymmetry that all future life would inherit. It's a captivating thought that the left-handedness of every protein in your body might ultimately be a relic of the polarized starlight that shone on a speck of dust in a nebula that existed billions of years before our sun was even born. Deep beneath the sunlit surface, shielded from the chaos of a young planet constantly pummeled by asteroids, another powerful engine was ticking away, providing a slow, steady, and relentless source of energy that could have nurtured life's first delicate steps. This theory turns our attention to the very atoms that make up the Earth itself, suggesting that the same natural radioactivity we often view with caution was in fact a vital creative force in life's origin story. Long before the first protocell learned to harness chemical energy, the planet's own radioactive heart may have been the power supply for prebiotic chemistry, offering warmth and activation deep within the crust where surface energy sources could not reach. The principle here is both simple and profound. Certain heavy elements like uranium, thorium, and potassium are inherently unstable. Their atoms spontaneously decay over immense timescales, shedding particles and energy in the process. This energy is released primarily as heat and radiation, alpha particles, beta particles, and gamma rays. On the early Earth, these radioactive elements were far more abundant than they are today, their concentration still high from the planet's violent formation. As water percolated through porous rocks and sediments in the deep subsurface, it would have been constantly bathed in this low-level radiation. Unlike the intense, destructive barrage of cosmic rays from space, this was a localized, persistent energy source, creating tiny, isolated hotspots of reactivity perfect for driving chemical evolution. The true magic of this process lies in what happens when this radiation interacts with water, the universal solvent of life. When an alpha or beta particle from a decaying uranium atom, for instance, tears through a pocket of water trapped within rock, it doesn't just heat it up. It ionizes the water molecules, violently stripping away electrons and creating a soup of highly reactive fragments. This generates what chemists call radical species, such as hydrogen atoms and hydroxyl radicals, which are incredibly eager to form new bonds. These radicals become the workhorses of prebiotic synthesis, swarming through the microscopic aquatic environment and slamming into any simple dissolved gases, like carbon monoxide, nitrogen, or hydrogen sulfide, that are present, forcing them to combine into more complex organic compounds. It's a form of chemistry that doesn't require light, heat, or mineral catalysts alone. It uses the direct, penetrating power of radiation to brute force molecules into existence. Laboratory experiments have brilliantly demonstrated the viability of this radioactive kitchen. Scientists have recreated these deep Earth conditions by taking simple mixtures of water, dissolved carbonates, and common minerals, sealing them in tubes with small, safe amounts of radioactive salts, and then letting them sit. The results are striking. Over time, these irradiated samples consistently show the formation of key biological precursors. Most notably, researchers have observed the synthesis of formic acid, acetic acid, and even the amino acid glycine, all driven solely by the energy of radioactive decay. These experiments prove that the complex chemistry of life doesn't require a lightning strike or a hydrothermal vent. It can proceed quietly and slowly in the dark, powered by the gentle, insistent tick of a radioactive clock, turning ordinary wet rock into a prolific organic factory. This theory provides an incredibly compelling environment for life's origins, particularly because it solves the problem of persistence. The surface of the early Earth was a hazardous place, subject to sterilizing impacts and scorching ultraviolet radiation. Any nascent chemistry happening in a tidal pool or a shallow lake could have been wiped out in an instant, but deep underground within the fractures of the crust, a community of molecules nurtured by radioactive decay would have been perfectly insulated. It was a stable, protected, and energy-rich refuge where slow, patient chemistry could proceed for millions of years without interruption, allowing for the gradual accumulation of complexity that eventually crossed the threshold into a living state. It suggests that life may not have been born in a dramatic flash, but in a slow, deep simmer, cooked by the enduring warmth of the planet's own radioactive bones. But what if the very flow of water itself, the relentless motion of streams and currents on the early Earth, provided a simple yet brilliant physical mechanism to bring order to the molecular chaos? Imagine a river carving its way through a primordial landscape, its waters rich with a messy jumble of organic molecules, short chains, long chains, charged particles, and neutral ones. In this turbulent mix, the chances of any two specific molecules finding each other to react are incredibly slim. This theory suggests that the solution to this problem wasn't a chemical catalyst, but a physical one, the natural sorting power of flowing water, a process that could have created pockets of concentrated potential, ready to ignite the chemistry of life. This concept, known as hydrodynamic sorting, relies on the basic principles of how different particles behave in a moving fluid. It's a phenomenon we can still observe today in any river or stream. If you've ever panned for gold, you've seen it in action. The heavier, denser gold nuggets sink to the bottom of the pan, while the lighter sand and silt are washed away by the water's flow. This same principle applies on a much smaller scale to molecules. In the gentle currents of a shallow stream or the capillary action of water seeping through porous sand, molecules don't all move at the same speed or in the same way. Their journey is determined by their size, shape, and electrical charge. Larger, heavier molecules, or those with a particular charge that causes them to stick to mineral surfaces like clay, will tend to lag behind. They get caught in eddies, trapped in small crevices, or simply settle out of the flow more quickly. Meanwhile, smaller, lighter, or more soluble molecules are carried farther and faster by the current. Over time, this relentless sorting action would have naturally segregated the primordial soup, creating microenvironments that were highly enriched with specific types of molecules. A quiet pool downstream from a turbulent rapid might become a rich repository for long-chain fatty acids, perfectly positioned to spontaneously form membranes. A tiny cavity in a rock, constantly fed by a trickle of water, could become concentrated with nucleotides, dramatically increasing the odds that they might link up to form a primitive RNA strand. Flow becomes a form of selection, not by biological fitness, but by physical properties, passively organizing the building blocks of life. This process offers an elegant solution to one of the most persistent puzzles of biogenesis, the concentration problem. The vast, dilute ocean is a terrible place for rare molecules to find each other, but a branching network of streams and rivers acts like a massive, planet-wide system of filters and concentrators. It takes a weak, scattered solution and actively processes it, focusing the valuable components into smaller and smaller volumes. The energy for this entire operation comes directly from the hydrological cycle, from rain, gravity, and solar heat, making it a ubiquitous and relentless force on the early Earth. It didn't require a special location or a rare event. It was happening everywhere all the time, turning every flowing body of water into a potential nursery for complexity, gently guiding randomness toward order through the simple, undeniable physics of flow. But to truly harness energy, a system needs a universal currency, a molecule that can store power and release it on demand for the countless tasks of living. In every modern cell, that currency is ATP, adenosine triphosphate, the energetic backbone of biology. But ATP itself is a complex molecule, a sophisticated piece of nanotechnology that couldn't have just appeared fully formed. So what powered the very first metabolic networks before this advanced financial system evolved? The answer might lie in a much simpler, more ancient class of molecules that are still vital to this day, polyphosphates. This theory suggests that long before ATP ever existed, life ran on a crude, mineral-based energy coin, and its echoes are still found in every one of your cells right now. Polyphosphates are, in essence, incredibly simple molecules. They are just long chains of phosphate atoms linked together by high-energy bonds, the same kind of bonds that give ATP its punch. Think of them as a string of firecrackers, where each connection between firecrackers is a little packet of stored energy, ready to be snapped to fuel a chemical reaction. And unlike the complex organic structure of ATP, these chains could form abiotically without any biological help at all. On the early Earth, rich sources of phosphate were likely present in certain volcanic minerals and within the structures of hydrothermal vents. The intense geothermal heat and pressure in these environments could have provided the energy needed to string individual phosphate units into these long, energy-dense chains, creating a natural probiotic battery waiting to be used by the first primitive metabolisms. The genius of this polyphosphate world hypothesis is how it provides a ready-made solution to the energy problem for any nascent life form. Imagine a simple iron-sulfur world protocell or an autocatalytic set clinging to a mineral surface. It has a network of reactions, but it needs a consistent spark to keep it going. A nearby source of polyphosphate minerals formed geologically could have served as that spark. These chains could have been broken down, releasing energy to drive otherwise impossible reactions, like building amino acids or forming nucleotides. This wouldn't require a complex enzyme. The mere presence of certain metal ions or even the slight acidity of the environment could have been enough to trigger the release of energy, making polyphosphates a universally accessible power source for early chemistry. The most compelling evidence for this theory isn't just in the past, it's alive and well in modern organisms. Polyphosphates are not some archaic relic, they are still produced and used by a huge variety of life forms from bacteria to humans. In our own cells, they play roles in energy storage, bone formation, and even cell signaling. But the real clincher is found in some of the most ancient and deep-rooted bacteria on the planet. Many species that live in extreme environments, like the hot springs and volcanic vents we've discussed, accumulate massive granules of polyphosphate within their cells. They use it as a compact energy reserve for surviving in harsh conditions, a clear echo of a time when this molecule was the primary and not just a secondary source of power. This widespread biological usage strongly suggests that the ability to harness polyphosphates is an ancient, fundamental trait, a cornerstone of metabolism that has been conserved for billions of years because it worked from the very beginning. It paints a picture of life seamlessly adopting a geologically available energy source and, over eons, learning to refine it into the more specialized and efficient ATP system we know today, all while never fully letting go of its original, powerful, and simple predecessor. While we often picture chemistry unfolding in watery soups or on solid surfaces, there's an entire realm of possibility floating in the air itself. This theory lifts our gaze from the primordial oceans and mineral scaffolds to the vast, open skies and the space between stars, proposing that some of life's most critical ingredients were forged not in a liquid or on a rock, but in the gaseous void. It's the concept of gas phase reactions, a fascinating process where the seemingly empty space of a planetary atmosphere or an interstellar cloud becomes a prolific chemical factory, building complexity from the simplest of atoms one collision at a time. The key to understanding this lies in the unique environment of a gas. In a dense liquid or on a solid surface, molecules are crowded together, constantly jostled and influenced by their neighbors. But in a low-density gas, molecules are isolated, drifting freely. While this might seem like it would prevent chemistry, it actually allows for a very specific and powerful kind of reaction. In the upper atmosphere of a planet or in the tenuous depths of a molecular cloud, individual atoms and simple molecules are constantly zapped by powerful energy sources, intense ultraviolet radiation from nearby stars, cosmic rays, or even lightning discharges. This energy doesn't just warm them up, it violently rips electrons away, creating highly charged, incredibly reactive fragments known as ions and radicals. These reactive fragments are desperate to regain stability, and in the emptiness of the gas phase, their only option is to find another partner to collide with. When they do, they can form new, more complex bonds in a single, rapid step. This process can create surprisingly large and intricate organic molecules. For instance, in the upper atmosphere of Saturn's moon Titan, which is rich in nitrogen and methane, UV radiation drives reactions that create vast haze layers of complex organic polymers called tholins, raining a constant shower of prebiotic material onto the moon's surface. This demonstrates that a planet's atmosphere isn't just a blanket of gases. It can be a dynamic chemical reactor, capable of manufacturing the building blocks of life from scratch and then delivering them directly to the surface below. Perhaps the most stunning evidence for this theory doesn't come from our solar system, but from the cold, dark clouds between the stars. For decades, radio astronomers have turned their massive dishes toward these seemingly empty regions of space, tuning into the specific frequencies at which molecules emit and absorb radiation. What they've found has completely revolutionized our understanding of cosmic chemistry. These interstellar clouds, far from being barren, are now known to be veritable soups of organic molecules. To date, astronomers have identified over 200 different molecular species in space. The list is nothing short of astonishing. They've found simple carbon chains, rings like benzene, and even direct precursors to life's machinery. In the vast Sagittarius B2 cloud near the center of our galaxy, scientists have detected the unmistakable spectral signatures of aminoacetonitrile, a direct precursor to the amino acid glycine. In other clouds, they've found ethanol, the alcohol we drink, floating in quantities that would fill countless billions of bottles. They've spotted acetic acid, the key component of vinegar, and glycolaldehyde, a simple sugar. These aren't just simple gases, they are the fundamental modules from which the much larger molecules of life are constructed. The fact that these complex organics can form in the extreme cold and near vacuum of space is a profound revelation. It tells us that the universe seems almost pre-programmed for chemistry, that the journey from simple atoms to complex molecules is a natural and widespread process. These gaseous factories in space operate on a cosmic scale, manufacturing the raw materials for life for billions of years. When a new star system forms, these pre-made organic molecules are incorporated into the swirling disk of dust and gas, becoming part of the recipe for every planet, asteroid, and comet that condenses out of it. So the gas phase reactions happening in the tenuous clouds between stars may have provided the universal starter kit, ensuring that any young, welcoming planet anywhere in the galaxy would be seeded with the same fundamental ingredients, ready for the next stage of assembly. And this brings us to a perspective that is perhaps the most profound shift of all, one that challenges the very notion of a single, triumphant originator. What if life did not begin with a solitary, self-replicating molecule winning a ruthless competition, but instead emerged from a collaborative effort? This is the theory of symbiotic origins, a concept that suggests the very first life was not a lone warrior, but a community, a cooperative network of different molecules and early protocells that found strength in partnership from the very beginning. We often view evolution through a lens of fierce competition, of survival of the fittest, but cooperation is just as powerful a force in nature, and its roots may run all the way back to life's very first moments. Imagine the primordial soup not as a gladiatorial arena where one molecule eventually dominated, but as a bustling marketplace of chemical possibilities. Within this chaos, there would have been many different types of simple protocells or autocatalytic sets, each with a tiny, incipient talent. One tiny vesicle might have been particularly good at absorbing energy from a nearby mineral gradient. Another might have, by chance, contained a molecule that could weakly catalyze the formation of a certain amino acid. A third might have had a slightly more stable membrane. On their own, these fragments of functionality were incomplete, barely clinging to existence. But if by chance they drifted together and merged, or if one engulfed another without destroying it, their strengths could have combined. The energy harvester could now power the catalyst, allowing it to work more efficiently. The stable membrane could now protect both of them. This synergy would have created a system far more robust and capable than any of its individual parts could ever be alone. This isn't competition, it's collaboration. The whole becomes greater than the sum of its parts, and this new cooperative alliance would have had a massive advantage, outcompeting not through brute force, but through superior organization and resilience. Life, in this view, may have found its foothold not through a singular discovery, but through the first successful handshake. The most stunning evidence for this theory isn't buried in ancient rocks, it's alive inside your own cells. We have a living relic of this ancient partnership, an event so monumental that it fundamentally reshaped the trajectory of life on Earth. This is the theory of endosymbiosis, and it tells a story that powerfully echoes the idea of symbiotic origins. Over 1 billion years ago, a large, complex cell engulfed a smaller, simpler bacterium. Instead of digesting it, a remarkable partnership formed. The smaller cell continued to live inside the larger one, and each provided something the other needed. The small bacterium was an expert at using oxygen to produce vast amounts of energy. The larger host cell provided protection and nutrients. This partnership was so incredibly successful that it became permanent. The swallowed bacterium lost its independence, and its DNA became incorporated into the host's own genetic machinery. It could no longer live on its own, and the host cell came to rely completely on its new internal power plant. That swallowed bacterium is what we now call a mitochondrion, the energy-producing organelle that powers every complex cell in your body, every animal, every plant, every fungus. The same process happened again with cyanobacteria, which were engulfed and became the chloroplasts that perform photosynthesis in plants. This wasn't a slow, gradual evolution of new parts. It was a sudden, revolutionary merger of two entire, functioning organisms into one. This event allowed for an explosion of complexity, leading directly to the vast diversity of life we see today. It stands as a powerful testament to the idea that collaboration, not just competition, is a fundamental and creative force in biology. This ancient story of endosymbiosis provides a compelling model for what might have happened at a much earlier, simpler stage. If entire cells can merge to form a new, more powerful form of life, is it so hard to imagine that the very first protocells did the same? The first partnerships wouldn't have been between sophisticated bacteria, but between loose collections of molecules and simple metabolic cycles. A vesicle good at concentrating protons could have merged with a strand of RNA that benefited from the energy gradient. A tiny metabolic network that produced a useful lipid could have been incorporated into a protocell that used it to strengthen its membrane. Each successful incorporation would have created a new, more stable platform upon which further complexity could be built. This theory suggests that the path to life was not a linear ladder with a single winner on each rung, but a sprawling, interconnected web where cooperation and synergy were the true engines of innovation, weaving simple chemistry into the rich tapestry of biology. But perhaps no image is more iconic in the search for life's beginnings than a lightning bolt cracking across a stormy, primordial sky. We've touched on the classic Miller-Urey experiment, but this theory deserves a deeper look with a modern understanding of where and how such awesome electrical power could have been most effectively harnessed. The vision isn't just of a single spark in a glass flask, but of a planet constantly alive with electrical fury, turning its entire atmosphere into a colossal chemical reactor. The raw power of a lightning strike is almost incomprehensible. In a fraction of a second, it can heat the air around it to temperatures nearly 5 times hotter than the surface of the sun. This isn't just a bright flash and a loud bang. It's a momentary creation of a plasma, a state of matter where electrons are ripped from their atoms, creating a superheated, incredibly reactive soup. When this happens in an atmosphere rich in gases like methane, ammonia, water vapor, and nitrogen, the effect is transformative. The incredible energy of the bolt shatters these simple molecules into their constituent atoms and radical fragments. Then, in the immediate, explosive aftermath as the superheated channel rapidly cools, these atomic pieces frantically recombine into new, more complex arrangements. It's a process known as shock synthesis, where chaos is forced into order by sheer, raw power. This is how the simple becomes complex, how a mixture of mundane gases can be forged into the very building blocks of biology in an instant. While Miller and Urey focused on the atmosphere itself, modern twists on this theory explore the specific environments where lightning's punch would have been most productive. One particularly compelling idea involves volcanic plumes. The early Earth was far more volcanically active than it is today, with countless eruptions spewing vast quantities of gases rich in hydrogen sulfide, carbon dioxide, and nitrogen high into the sky. These ash-rich plumes colliding with the humid atmosphere would have been perfect for generating intense, frequent lightning strikes, a phenomenon we still observe in volcanic eruptions today. But the key is the location. A lightning bolt within a volcanic plume is doing its chemistry right inside a concentrated cloud of the very ingredients it needs, and the resulting organic molecules would then rain down directly onto the Earth's surface, often landing in the mineral-rich, watery environments around the volcanoes themselves, ready for the next stage of assembly. Another modern perspective focuses not on the sky, but on the point of impact. When lightning strikes the ground, it doesn't just vanish. It can fuse sand into beautiful, glassy structures called fulgurites, literally lightning fossils. But the chemical effect might be even more important. A strike on a beach or shallow seabed rich in certain minerals could have done more than just provide energy. It could have provided a catalyst. The immense heat and pressure at the point of impact could have created temporary, localized conditions that dramatically accelerated specific reactions, perhaps even helping to form those tricky nucleobases or sugars that are so essential for genetics. So the lightning bolt isn't just a spark in the sky, it's a precise hammer blow, striking anvils of particular minerals to forge specific molecular tools. This paints a picture of the entire planet as a grand workshop, with lightning as the universal power tool, actively and relentlessly manufacturing the pieces of the puzzle wherever the conditions were just right. Yet even after 30 different theories, from the depths of the ocean to the far reaches of space, we are forced to confront one last staggering possibility. What if the origin of life wasn't just unlikely on our planet, but was so profoundly improbable that it required not just one universe, but a near infinite number of them to succeed? This is the ultimate cosmic perspective, the theory of multiverse life, a concept that suggests our existence is only possible because we find ourselves in one of the rare universes where the dice of reality finally rolled in life's favor. To understand this mind-bending idea, we need to grapple with the anthropic principle. In its simplest form, the anthropic principle observes that the universe we observe must be compatible with our existence as observers. It sounds almost trivial, but its implications are anything but. When we look out at the cosmos, we see a universe that seems almost perfectly fine-tuned for life. The fundamental constants of nature, the strength of gravity, the mass of an electron, the force that holds atomic nuclei together, appear to be exquisitely balanced. If any of these values were even slightly different, atoms wouldn't form, stars wouldn't ignite, and the complex chemistry necessary for life would be utterly impossible. The universe would be a lifeless void, so why is everything set just right? The multiverse theory offers a radical answer. There isn't just one universe with one set of rules. Instead, there is a vast, perhaps infinite, ensemble of universes, often called the multiverse, each with its own unique set of physical laws and constants. In the overwhelming majority of these universes, the constants are misaligned and life never gets a chance. But in a tiny, tiny fraction of them, the numbers line up perfectly, and we shouldn't be surprised to find ourselves living in one of those rare Goldilocks universes where everything is just right. Now apply this same logic not to the universe's laws, but to the specific event of life's genesis. We've spent this entire journey exploring how difficult it is to bridge the gap from non-living chemistry to a self-replicating, evolving system. Every theory we've discussed faces significant challenges. The steps seem so numerous, the hurdles so high, that some scientists and philosophers have argued that the probability of life emerging by natural processes alone, even in a universe with perfect constants, is astronomically low. This is where the multiverse theory takes a breathtaking leap. It posits that within this vast cosmic landscape of parallel realities, the spontaneous emergence of life is happening constantly. In most universes, it fails. Protocells never form, RNA strands never replicate, metabolic networks never achieve closure. But in an impossibly rare subset of universes, through sheer statistical inevitability given enough trials, every single one of those unlikely steps somehow falls into place. In those universes, life blossoms, and of course, we can only be in one of those universes, marveling at our own existence, because in all the others, there's no one around to wonder about it at all. This idea transforms our search for life's origin from a historical puzzle into a cosmic necessity. It suggests that the question, how did life begin, might be less important than in what kind of reality can life begin. The answer, according to this view, is a reality of incomprehensible scale and diversity, where every possible event, no matter how improbable, must eventually happen somewhere. The first living molecule on Earth wasn't just a lucky accident on one planet. It was a statistical inevitability in a cosmos of infinite planets within infinite universes. This doesn't diminish the wonder of life, it expands it to a truly cosmic scale, framing our existence as a single, shining day to point in a reality of limitless possibilities. It is the final frontier of origin theories, not because it provides a mechanism, but because it provides a context, a reality so vast and strange that the miracle of life becomes, in a sense, ordinary. From the protected depths of Earth's crust to the frozen, silent hearts of comets sailing between the stars, the search for life's origin is a tapestry woven with countless incredible threads. We've ventured through 30 distinct theories, each proposing a radically different setting for that first miraculous spark. We've witnessed life potentially emerging from the chaotic energy of a lightning strike, the patient, rhythmic pulse of tidal pools, the gentle magnetic guidance of the planet itself, and even the statistical inevitability of a multiverse. We've considered the possibility that our most distant ancestors were not mere molecules, but interstellar dust grains or even intentional emissaries from an advanced civilization. This isn't a story with a single, simple answer. It is the grandest of scientific mysteries, and every theory, from the warm little pond to the cold clay catalyst, adds a vital and fascinating piece to the puzzle. What makes this exploration so profoundly compelling is that it's far more than a historical question. It's a question about our very identity and our place in the cosmos. Each theory forces us to see our existence through a different lens. Do we owe our lives to the unique geology of our planet, a native product of Earth's dynamic systems, or are we citizens of the galaxy, the result of a cosmic diaspora that seeds life across star systems? The answer shapes how we view everything from the resilience of life to the very likelihood of finding companions among the stars. This search for beginnings is ultimately a search for meaning, challenging us to decide if life is a singular miracle or a universal imperative. Now we turn this incredible question over to you. With 30 captivating possibilities on the table, which one resonates most deeply? Does the elegant simplicity of the RNA world feel like the most likely path? Or does the profound idea of directed panspermia, that we might be part of a grand, intentional experiment, capture your imagination? Perhaps the protected, energy-rich environment of the deep Earth biosphere strikes you as the perfect cradle, or the clever physics of hydrodynamic sorting seems like an unsung hero. Dive into the comments below and share your thoughts. Tell us which theory you find most compelling and why. Let's continue this conversation together, because the mystery of how we got here is a story we are all a part of. If unraveling the cosmos's greatest secrets fuels your curiosity, you're in the right place. We're just getting started. For more deep dives into the wonders of space, the puzzles of physics, and the enigmas of existence, make sure to hit that subscribe button and ring the notification bell. The universe is full of mysteries waiting to be explored, and we can't wait to journey through them.